Hello, everyone. Welcome to the ISCT webinar, Qualifications of Ancillary/Raw Materials for Clinical Use. We are proud to present today's webinar with the support of STEMCELL Technologies and the ISCT North America Legal and Regulatory Affairs Committee. My name is Deborah Griffin, and I will be the chair for today's session. Today's presenters are Lynn Santos and Anna Gilbert. Lynn Santos is the Senior Director of Quality Assurance and Regulatory Affairs at STEMCELL Technologies. She has worked in the biotech industry for over 20 years, transitioning from chemical development into quality assurance and regulatory affairs approximately halfway through her career. Although her background is primarily with small molecule API and drug products, she has been able to leverage her knowledge of the regulations surrounding small molecules, peptides, and lipids to navigate the regulatory landscape that is currently being defined for cellular therapies. Working at STEMCELL Technologies since 2008, she's had the opportunity to witness firsthand the ever-changing compliance requirements as they relate to regenerative medicine, and is excited to be part of an organization that is helping shape the future landscape of this emerging field. Anna Gilbert has over 20 years of experience in quality assurance and biotechnology, with the last 8 years focused in cell therapy. Anna is currently the Director of Quality at NOLA Therapeutics Incorporated. Prior to NOLA, Anna has prior experience at several biotech companies. Most recently, she started in January 2014 at Juno Therapeutics as one of the earliest employees and the first in quality assurance. She established the first quality systems for Juno and managed the GMP quality compliance program. At Dendrian, Anna managed all aspects of GMP and GTP quality compliance, including internal and external auditing, as well as compliance oversight of apheresis sites. She joined Dendrian in 2009 and participated in the successful pre-approval inspection of the first U.S. commercial cell therapy, Provenge. During her tenure at Amgen, she supported quality operations for manufacturing, quality control, validation, and new product introduction for clinical phase monoclonal antibodies. Anna had the good fortune to arrive at Immunex in 1998, just prior to the launch of Enbrel, at the time the most successful biotech product launch. Anna began her career at Chiron, supporting the lot disposition of diagnostic kits for blood screening. Anna is an ASQ certified quality auditor and a member of the Parental Drug Association. She delivers an annual lecture on the topic of GMP auditing at the University of Washington's Master of Science in Biomedical Regulatory Affairs program. I now welcome Lynn Santos to present.
Thank you. Thank you, everyone, for joining us today. I will start by talking about what the topic, which is ancillary materials and raw materials. So these are materials that are used as components in the manufacture of biological products, such as cellular therapies. And they are important substances that are used as part of your protocol, but are not intended to be part of your final cellular therapy. A great guideline to reference is the USP 1043 titled Ancillary Materials for Cell, Gene, and Tissue Engineered Products. This particular chapter of USP provides a great guiding framework to qualify ancillary materials. It's important to note that when you are looking at your protocol, that you consider your entire workflow. So often we get focused on areas such as the specific reagents that we use, but it is also important to consider the filters, the packaging, and anything that comes in contact with your final therapy, cellular therapy. So you need to consider your entire workflow as in the context of USP 1043. It's also important to note that the GMP stringency increases as your product moves through the clinical phases to commercialization. So when you're starting out with your preclinical, it is often the compliance required for those early phases of your translational journey is often less than what is required for phase 3 and commercialization. And some of these differences lie within the validation requirements. So it's important to have these conversations with your suppliers of your raw materials and ancillary materials to ensure that they are on a similar journey as you are to ensure the appropriate compliance as you move towards commercialization. So what are the qualification expectations? So in essence, regardless of what regulatory authority you are dealing with, they expect some minimum requirements. The first is to qualify the raw materials, and they expect consistent control. So that means that you need to ensure that every time that you order that particular material, it is going to be the same and will not impact your particular protocol adversely. They expect you to use the best quality available and the materials that work for your application. So GMP, unfortunately, is not always readily available from your suppliers, but that doesn't mean that you cannot use them. But if there is the exact same product that works exactly the same and one is GMP and one is not, the regulatory authorities will likely expect you to use the GMP grade. You need to know your material so that you can defend its use for your application. And it's very important to know that the claims from suppliers are not standardized. And sometimes suppliers can overstate what their compliance is. So you see messaging from different suppliers such as GMP grade, clinical grade, or following applicable GMP guidelines. It's important to vet what that means with your supplier and don't blindly accept the claims that they make. So I'm going to take you through a step-by-step guide of how to qualify your materials. So step one, you need to determine if the ancillary material works for you. If it doesn't work for your application, it doesn't matter how compliant it is because it will not be applicable for your use. So it's important that you test it and then work with your supplier to gain understanding of its compliance. So some of the questions that you need to ask yourself are, do you get the isolation you need or the purity that you need? Or do you get the expansion from the media that you need? So it's important to, first of all, test the material. Step number two, determine if the supplier can support you. So some of the questions to consider is, has the supplier supported previous clinical trials with their products? Do they have a reputation of supporting the translational journey for cellular therapies? What is their compliance roadmap? So perhaps the material that you are leveraging for preclinical and phase 1 is not GMP, but does the supplier have a line of sight of getting that material to GMP by the time you arrive at phase 3 and commercialization? What is your relationship with me? I say me because I am representing a supplier, but that is very important to build that because you are going to be in this relationship over anywhere from 2 to 5 to beyond years. And that means that you're going to need to work very closely together with your supplier to ensure that they can support you along the way. Are they responsive? And do they have the documentation you need for your clinical submissions? Number 3, you need to gather the documentation to support the compliance claims. So back to the earlier slide where you cannot blindly accept the compliance claims, you need to actually verify them. So you need to ask for things like a certificate of analysis and take a look at it and determine, does it meet your needs? Is there enough information on there to support your submission? A certificate of origin, are the claims to primary or secondary levels? What do you need? And depending on the jurisdiction to which you want to run your clinical trial, these are critical questions. Do they have a reference to a DMF or a master file? This is very desirable, but it is not always available. Do they have stability data to support the shelf life for the use in your protocol? And you need to be assured that the material will not have a negative impact on your product or the patient. So that is critically what the regulatory authorities are looking for. And I just wanted to dedicate one slide to what drug master files are. So the purpose of a master file are threefold. So there's I broke it down into 3 different perspectives or lenses. So from a supplier's perspective, it's a way for us to protect our trade secrets. This is a place where we put the product formulation, all of the information that the regulatory authorities may need to assess the safety of the ancillary material. The regulatory authorities, again, they will look at the DMF if it is referenced within an IND, a BLA, or an NDA. And to the sponsor, which is the customer, this provides support to their translational pathway. So you need to know specific information about the ancillary material to effectively justify its use. So not all jurisdictions have master files in place or a process in place, and so what we have found as a supplier is that we may provide ad hoc information to regulatory authorities to support the sponsor's submission. So you can work separately again with your supplier to make that happen. Number 4, you need to confirm the documentation. So step number 3 was to request the information, but again, don't take anything for granted. It is up to you to justify the use of this material and the safety of this material for your application to the regulatory authorities. So it is recommended that you conduct testing as per the C of A for a minimum of 3 lots and probably at least 1 lot year after year after that. Check for lot-to-lot variability. Ensure that the supplier is reporting accurate results, and characterize the testing of the ancillary material and set specifications that meet your protocol. So this is, again, a conversation that you need to have with your supplier to assure that the specifications set for the ancillary material meet your requirements. And really, from your perspective, not the supplier's perspective, what are the critical quality attributes of the ancillary material as related to your protocol? Step number 5 is to conduct a risk analysis. So consider where in your process the ancillary material is used and what are the chances of carryover of the ancillary material into the patient. So we work with customers that use our reagents or ancillary materials very early on, for developing such things as master cell banks. But we also work with customers that use our ancillary materials directly before injecting the patient with the cells. So there's a risk-based approach, and the compliance requirements for each of those examples may differ. And you always need to consider the patient, and that is because that is the number 1 area of risk that the regulatory agencies are looking for. Does the ancillary material have animal components? And determine if the lot-to-lot biocompatibility, cytotoxicity, or adventitious agent testing is needed. So this will be dependent on what the general components are of the ancillary material. Number 6 is audit the supplier. So sometimes we like to just believe that the supplier is functioning out of a state-of-the-art facility and producing results using qualified procedures and processes, but it's very important for you to make sure that this is actually true. And a good measure always as an experienced auditor is to take a walk through the facility and to determine its cleanliness and orderliness. And of course then take a look at the documentation to support all of the critical documentation that you are leveraging for your submission. Step number 7 is to put a quality agreement in place with you and your supplier. This helps define exactly what you expect the supplier to do for you. As you're building that relationship, and as you know, even with communicating on a regular day basis, it's not always clear that the expectations are aligned between 2 parties. So writing this out in a document is something that is very standard in industry, and it captures things such as change notification, what your specifications are for the ancillary material, how often you're going to audit them, and any other critical aspects to ensure your success. Number 8 is assessing the removal of ancillary material from your final product. So this is a bit tricky as we know that the science is not all necessarily there to measure small amounts of an ancillary material that still may be left on a final cellular product. But the agencies do expect you to develop testing methods to assess what is there, what has been removed, and maybe some testing has been done by the supplier to develop this, and maybe they've generated data, but even if they have, you need to repeat this testing within your specific protocol because each customer's use of a particular ancillary material is different. And here is an overall chart that is really a summarization of what is outlined in USP 1043. And this divides, first of all, it lists the qualification activities that I've just gone through in the 8 steps, but it also assigns accountability. And as you know, supplier does have accountability to, is part of that relationship that you're building over your journey. But the end user, the sponsor of the clinical trial, has an enormous amount of the accountability because you are the ones that are going to be facing the regulatory authorities and giving the justification of the use of this particular material. So it's important to note that there is no one-size-fits-all. Each qualification plan is going to be different. It's going to be based on different variables, such as whether the ancillary material has animal components, where in the protocol the ancillary material is used. Is it close to the patient or far away from the patient? So each of these qualification plans needs to be created through your relationship with your supplier. And just a note, we had published a white paper through Cytotherapy a few years ago, and this particular white paper goes into a lot more details about the regulatory landscape and the qualification requirements. So please look that up if you have the chance.
Thank you, Lynn. I now welcome our second speaker, Anna Gilbert.
Thank you. For my portion of the webinar, I will build upon Lynn's foundation with the 8 qualification steps for ancillary materials. While I will not focus on one specific material, many of the examples I will use are based upon cell expansion media with biological components. I will also include other real-life examples and points to consider that may not be directly related to cell expansion media. The first step is to determine if the ancillary material works for you. The transition from the bench or academia is often based on short-term deliverables instead of long-term deliverables. The number one short-term deliverable is typically speed to clinic and to commercialization. The long-term deliverables that may be overlooked are whether the supplier can provide adequate amounts of material based on projected need and lead times for material delivery. An evaluation of risk related to animal-derived materials is typically not performed during early development. Cells grow well in the presence of animal proteins, and looking to future global regulatory requirements is often not considered. Finally, cost of goods, or COGS, is not a priority in early development. Purchasing a small quantity of an expensive item may not be an issue early on, but as the process scales up, this may be prohibitive to commercialization. In some instances, suppliers may provide price breaks for academia that are no longer available as the process moves towards commercialization. The relationship with service providers and suppliers is critical. While legal agreements are important to defining the relationship, there is no substitute for communication. A strong relationship with a supplier can pave the way to ensure the material is appropriate for later phase and commercial product, as well as expansion to other regulatory jurisdictions. For example, you may need to work with your supplier to ensure all components of the media are compliant with countries you are seeking to expand into. Japan is very specific about understanding the inputs and outputs of each product and process. For the inputs, the PMDA wants to know everything about the raw materials, in particular those of human or animal derivation. For media that may contain insulin, you should know if the insulin manufacturing process contains porcine-derived trypsin for cell dissociation. If it does, you will need to obtain documentation that the trypsin has been appropriately tested for adventitious virus as per 9 CFR requirements for ingredients of animal origin used for the production of biologics. Alternatively, your supplier may be able to obtain recombinant trypsin for your media. Another component that is thoroughly reviewed by jurisdictions such as Japan and the EU is bovine serum albumin, or BSA. As an example, Japan may seek specific information for BSA, such as verification that it was manufactured after the date the country of origin was recognized by the World Organization for Animal Health, the OIE, as negligible risk for BSE. For the U.S. source BSA, this date is May 29, 2013, so you must be able to work with your supplier to ensure the collection date is compliant with the OIE. It is also important that you can obtain information for the viral inactivation and viral removal capability of the BSA included as a component in your media. Without a good relationship with your supplier, this information can be difficult or impossible to obtain. Also, keep in mind that some companies may provide some of this information on their website. Some suppliers may not be willing to supply their material for processes that are late stage or commercial. You must verify with your supplier that they will provide their material for all phases of your process. Another potential roadblock in the use of materials is licensing agreements. In 2013, Life Technologies exclusively licensed Dynabeads to Novartis. Dynabeads are used to isolate, activate, and expand therapeutic T cells for use in CAR T therapies. Dynabeads were used by many early phase processes, but companies needed to switch to other reagents to avoid licensing issues. It is important to verify that your supplier can supply all the necessary documentation to ensure the material is of consistent quality and safety. The C of A may have all the assays and acceptance criteria you require. However, the C of A may not have all that you require, and in that case, you will need to negotiate with your supplier to add more testing, or you may need to do supplemental testing on your own. Supplemental testing paid for by you should also be considered when it comes to cost of goods. One supplier may provide a better price for a material but have inadequate testing reported on the C of A. The cost of the supplemental testing may cause that supplier to no longer be the best provider of that material. For animal-derived materials, particularly bovine, you will want to ensure you can obtain a certificate of origin. This document should include the country of manufacture, age of the animal, verification the abattoir is USDA licensed for U.S. manufactured serum, and the animals were inspected ante and post-mortem. The supplier may also be able to give you a certification of suitability to the monographs of the European Pharmacopoeia, or CEP. CEPs are recognized by the signatory parties of the Convention on the Elaboration of a European Pharmacopoeia, that is, all member states and the EU. They are also recognized by other countries, for example, Canada, Australia, New Zealand, Tunisia, and Morocco. A CEP document will provide verification that the serum complies with the European Pharmacopoeia. Your supplier must also be able to support stability and expiration of its products so you can be assured of consistent performance in your production process. If your supplier cannot provide you documentation for the storage and shelf life of your material, you will need to perform your own stability studies. As with supplemental testing, this can impact your cost of goods. Stability testing can be lengthy and expensive. It is important to verify the appropriateness of the test methods used by the supplier to test the material. For example, the sterility assay must be fully validated to provide true assurance of sterility. Safety assays are required to be fully validated in early phase clinical trials. The supplier may not have performed growth inhibition studies with your particular formulation. You should be able to work with your supplier to achieve a fully validated sterility method with your formulation. Be aware there are one-time costs associated with this. In addition to the appropriateness of test methods to provide sterility assurance, you may also be able to work with your supplier to mitigate gaps in the manufacturing process. A manufacturing process for media will not include terminal sterilization techniques such as autoclaving or sterilization via radiation, as these methods may negatively impact functionality of the media. As such, media are typically 0.2 micron filtered. To provide assurance the sterile filtration was successful, a post-filter integrity test should be performed. If it is not, you cannot be assured the filtration provides appropriate sterility claims. You may work with your supplier to perform a post-use integrity test for each lot produced. This will be an additional cost that will impact each lot. This example demonstrates why you should not take the assays and acceptance criteria reported on a C of A at face value. Any animal or human-derived material you include in your process must be assessed for risk. Risk for these materials may be tied to parameters such as quantity used and where used in the manufacturing process, the robustness of analytics to detect the presence or clearance of those materials. Assessment of clearance applies to components of your material. For example, a cell expansion media may be evaluated for the clearance of BSA, insulin, and transferrin. Many jurisdictions may also want to know about animal-derived materials and possibly viral validation for recombinant raw materials that may be derived from microbial or mammalian cell lines. It is helpful to have a strong communication with your supplier to ensure you can either obtain this information directly from them or know that they are able and willing to contact regulatory authorities directly. Many suppliers are not able to disclose proprietary ingredients or processes related to their material. In those cases, you must ensure there is a master file to reference or that their regulatory group is willing to directly contact and disclose to a regulatory authority. For direct contact with a regulatory authority, you will need to provide your supplier with the regulatory authority reviewer name and contact information. Animal or human-derived materials must have documentation to demonstrate that all viral testing has been performed per regulations. If all required testing hasn't been completed, it is upon you to close the gap with additional testing. While it is important to have a strong and open communication with your supplier, it is imperative to perform a site audit of your supplier. The audit is what will help you to have a full understanding of the manufacturing, testing, and compliance of your material. An example of a manufacturing process to understand is how the final lot is made. Some companies may formulate a large parent batch that is stored long-term and then thawed, filled into a final container, labeled, and shipped. This is sometimes called a sales lot. This is important to know because it may impact the amount of testing you do for each sales lot, as you may be able to leverage testing from the parent lot. Other companies may complete the fill at the time of formulation and relabel for each sales lot. Again, this is critical to know, as you may have risk associated with labeling. You also may find some things that can be startling, such as the use of antibiotics in the same facility as where your material is made. Process and facility flows can be seen and assessed for robustness and the ability to control cross-contamination. You should also have the opportunity to see personnel performing tasks, which will provide the ability to assess appropriate gowning, training, and technique. And you will be able to assess the facility for cleanliness. You can look for rust, water stains on the ceiling, and evaluate the cleanability of the area, including non-porous surfaces and coved flooring with no gaps. A quality agreement can be a standalone attachment to a master services agreement, or it can be embedded within the MSA. The quality agreement will enable each party to understand who completes tasks such as lot disposition and testing. You will want to ensure that critical items such as communication, scope, and timing of changes is clear. For example, a change to a component in your media may greatly impact your product, and your firm will need time to perform process development activities. This should be clear in the quality agreement. Other critical items include deviations and investigations, out of specification, right to audit, and communication of regulatory inspections. As the sponsor, you are responsible to know and understand your process capabilities, including the removal of ancillary materials from the final product. To assess ancillary materials removal, you must first determine and develop the appropriate analytical methods, such as an ELISA-based method. Once you have developed an appropriate analytical panel, you can now assess and quantify the removal of materials used in your process. Regulatory authorities require analytical data to show removal. A theoretical calculation may be used as supporting information, but it is not sufficient on its own. The removal of ancillary materials is specified in USP 1043, Ancillary Materials for Cell, Gene, and Tissue Engineered Products. Quote, ancillary materials are not intended to be present in the final dosage form in cell, gene, and tissue engineered products. Their presence in the final product could lead to undesired effects in the recipient or have a detrimental effect on product potency. Additionally, these risks can be mitigated through the design of processes to include steps to adequately remove the material through dilution, separation, or inactivation, as well as the development of analytical detection assays to assess the material levels during processing and in the final therapeutic product. Assessment and removal strategies for residual materials should be considered early in the process development phases. There are two different approaches for assessing residual material levels in the final therapeutic product. One, validation studies can demonstrate that the process is capable of removing more of the material than would be present in the worst-case scenario. Or two, the residual levels of a material can be measured for each lot at an appropriate step in the manufacturing process. For complex ancillary materials, such as media, you will need to assess each component for residual in the final product. Thank you for your attention.
Thank you, Anna. That concludes the presentation segment of the webinar. We will now begin our Q&A session by addressing the written questions that were submitted throughout the presentation. After we have answered these, we will take questions by phone. So our first question is for Lynn. What do you mean by primary or secondary levels with C of O?
Okay, so a primary material is considered to be the raw materials used directly in the manufacture of the ancillary material. Secondary level of traceability are the materials that are used to manufacture the raw materials of the ancillary material. And maybe I could ask Anna to give a specific example.
Yes. So the primary material may be something, as I discussed, like a cell expansion media, and the secondary component could be something, as Lynn mentioned, insulin, which is a component in the media.
And in reality, there could be a tertiary level as well, right? Because it turtles all the way down. You have to go back to the source material, the primary source of the. I guess, is it an API at that point? No, it's the raw material.
It's just a raw material. So it's basically, you can look at it as just layers that you have to dig down to the touch point. So what went into the manufacture of the expansion media, and then you find out that one of the components is insulin. So that is a primary raw material. But then how was the insulin manufactured? Did it touch any animal components in the process? And so that would be the secondary level.
Thank you. Our second question is, how long does the qualification process usually take? And Anna, I'd like you to address this. And what is the longest part of that process?
That's a great question. So there's kind of two portions I see as part of the qualification process. One is the actual qualification of your material, and Lynn talked about that a little bit in her portion of the presentation. So qualifying the material, making sure that it works in your process and that it can be adequately cleared from your process, and also that each lot manufactured is robust and reproducible, so there's not a large amount of variability from lot to lot from your supplier. And then the other portion of qualification would be more of the supplier qualification. So working directly with your supplier to perform a site audit, verify that you can obtain all of the appropriate documentation from them. So I would guesstimate 6 to 9 months for the entire process.
Thank you. The next question is, where do people typically get stuck, and what are the common bottlenecks? Anna?
Let's see, where do people typically get stuck? Well, there's probably a million different examples, but one example that I have seen is you have a research grade material that performs really well in your early process development, and either the higher grade material doesn't perform as well in your process, or there isn't a higher grade material. So trying to determine whether or not you can find another material that's appropriate, or determining whether or not you can move forward in regulatory jurisdictions with that material, that can definitely be an issue and a bottleneck.
And I think part of that is the expertise of the supplier that you're dealing with as well. You should definitely reach out to your suppliers to help you with getting through those bottlenecks.
Yeah, agreed.
Our next question is, if my use of the material is for a different application than what the supplier advertises, is it important for me to conduct my own stability studies? For example, if I am expanding a different cell type than the cell type the supplier's media has been tested for. Lynn?
Yes, that's a great question, and definitely even if you are using it for the same application, it's still in your best interest to verify the stability data, either through an audit of the data to ensure that it meets the compliance requirements such as the ICH guidelines. But if you are using it for another indication, it is definitely, again, in your best interest to conduct those stability studies yourself to ensure that the material does not adversely affect your protocol or the outputs of your cellular therapy.
Great, thank you. The next question is, are there standard supplemental tests, a standard battery of tests for the certificate of analysis? Anna, can you address that? In your experience, have you do you test a standard set of parameters when you're looking at the C of A's and deciding that that's not quite enough?
I'll give you my standard answer, which is it depends. So certainly some materials, the testing that's required on them is dictated by potentially the Code of Federal Regulations, ICH, USP, or EP. So certainly those can be leveraged. From there, it depends on how and where you use it in the process and what parameters of that material are critical to your product performance and your product quality.
Thank you. Our next question is, what does a supplier audit typically look like? And I'd like each of you, Lynn and Anna, to answer that because, of course, it's from two different sides. Lynn, can you go first?
Yes. So STEMCELL and myself actually enjoy getting audits from our customers because it's an opportunity to build that relationship out of the gates. And there's nothing that is better than face-to-face communication and understanding what each person's or each company's requirements are. And it is also an opportunity for the company to continually to improve our quality management system as a whole. And we often definitely include a facility tour. We often try to focus it from the receipt of raw materials for our manufacturing purposes all the way through production, quality control testing, and then through to the final packaging and release of the particular ancillary material. And this is followed by an in-depth look at the documentation that support essentially that tour and the things that we proclaimed on that tour. So it's definitely a great opportunity, I think, to highlight all the good work that you do as a supplier, learn more about your systems from a new perspective, and also to build that relationship.
Great. Anna?
So I would definitely echo what Lynn said about starting off with the facility tour, although usually you have an opening meeting with both the customer and the supplier, and I find that it's helpful to have a brief presentation to show how their material fits into your process, because it can definitely help to frame conversations like why you might find a particular parameter to be important or why you might ask questions, and it also helps the supplier to know how their material is being used. So I definitely encourage that. And I always like to start my audits off with a tour and just basically follow the entire process flow, starting from material receipt and release in the warehouse area, looking at how materials are received into the manufacturing area, how QC samples get out of the manufacturing area, having a chance to actually see technicians at work, have the ability to view their aseptic technique and assess whether or not you believe they've been appropriately trained, and then also see how the product or material gets out of the manufacturing facility and then moves into final labeling and packaging. So it's helpful to see everything from start to finish. And as Lynn and I both discussed, it's a good chance to actually see the facility and make sure that it's maintained appropriately. Facility cleaning is absolutely imperative to ensure that your material has not been contaminated. Also evaluating material, personnel, and waste flow throughout the facility, and that's something that you really cannot assess unless you're there in person. And then, as Lynn said, following the tour, typically you do a deep dive into documents to verify that the supplier has appropriate systems in place and that they also follow those systems.
Great, thank you. Our next question is, when is GMP absolutely needed? I'd like you both to address that. Lynn?
Yeah, so we have discovered as a supplier that this varies by region. So we find that in the United States and North America that this is somewhat flexible, that the FDA is really looking for that robust qualification plan of the material, and they aren't necessarily looking specifically at whether or not it's GMP grade or not. They don't use that as a black and white go, no-go measure. However, as you progress through the different phases of your translational journey, the expectation of compliance increases, and we are, through experience, STEMCELL is determined that GMP is likely required in late phase clinical trials, so phase 3 or even, and definitely, absolutely by commercialization. So sometimes even for phase 3, we're finding some flexibility if the indication of the clinical trial is for an orphan indication. And again, the FDA shows some flexibility, still very much expecting a robust qualification plan to ensure that they are not introducing undue harm to the patients. However, again, just noting some flexibility there. But in comparison, we note in Europe and other jurisdictions that the regulatory authorities expect GMP very early on, even back into preclinical, as we have gained feedback from the MHRA. So when we ask them specifically, what happens when there is no GMP grade readily available on the market, they replied that they too would expect a qualification plan and a justification of why that material is being used for the particular application. So there appears to be less flexibility in jurisdictions such as Europe.
And I would definitely echo what Lynn just stated. So I would say a rule of thumb is when you get to phase 3, that is definitely when you want to be pursuing higher grade or appropriate grade of material. And one of the reasons for that is as you move into your phase 3 clinical trial, that is the clinical data that you're using to support commercialization. And if you change materials throughout that trial, you may need to do additional comparability studies, which can take a long time and also be costly. So I would say rule of thumb is when you're heading into phase 3, do a really close look at all of your materials. Make sure that you are obtaining the best possible grade. And as Lynn said, where there may not be GMP grade available, that you're doing an appropriate assessment of those materials.
And also something I noted during Lynn's talk is that she spoke about a compliance roadmap, and does that supplier have a plan to continue to refine that product and develop it? So I think that's important to ask as well, which I actually hadn't considered previously. So our next question is directed to Lynn, and do you get audited by academic institutions who are using your reagents for clinical trials?
That's a great question. Thus far, no. It appears, and this is an opinion, okay, make that very clear, based on the experience that we've had, and so not necessarily a fact out there, but it appears that there's more flexibility in startup clinical trials when dealing with academic institutes compared to small biotech companies. So again, this is an opinion. So we have noted through supporting over 30 clinical trials around the globe that the academic institutions don't or have not come audit us to date, even as they have progressed through phase 2 clinical trials. However, small biotech or industry customers are the ones that definitely come audit us.
Thank you. Our next question is also directed to Lynn. Lynn said at the start of her presentation that consumable-derived ancillary materials are available. Can you please provide an example?
Yes. So maybe I misspoke around how I phrased that. So consumable-derived ancillary materials. What I actually meant to say, if that is indeed what I said, that there are consumables that are considered to be ancillary materials. And these are things such as filters that you might be filtering your final cellular therapy product through, or they could be your final container closure of your cellular therapy. So those consumable items that you assume would be inert and appropriate for use, you still have to qualify them in the same manner as you would for a media or a cell isolation product.
Yeah, I remember for one of our INDs, we had to qualify the filter to prove that it was a non-shedding material and did not contaminate the cell therapy product. So yes, that's important. Our next question is directed at Anna. I guess, is there standard language for service agreements that you can recommend?
That's an excellent question. Just based on my experience, it seems like maybe there was one lawyer at the beginning of it all that started some boilerplate that seems to circulate around. So I'm guessing we've all seen pretty much the same template. But as far as standard language, I would frame it more that there are standard things that you want in your agreement. And for me, I'm in quality assurance, so particularly around quality. So, having in your agreement that the supplier does and is required to have appropriate systems in place, such as personnel training, a document control system, CAPA and deviation system, a change control management system, and then also a robust material receipt and release system. So those are certainly things that I would expect that you would want to have in your service agreement.
And this is Lynn. I'll just add a couple comments to that. And change notification is clear, so having a change control system in place, but it's very important as a customer to ensure that your supplier isn't changing, the ancillary material in a way that would impact the performance in your protocol. So I think that clarity is good to call out in the quality agreement. And the FDA actually has a guidance. I don't know the exact title, but if you query FDA guidance and quality agreements, you will come up with the guidance there, and they provide some framework of how to put these agreements in place.
Great, thank you. That concludes the end of the text questions. Thank you for joining today's webinar. If you have additional questions, please email them to ict@celltherapysociety.org. A webinar evaluation will be sent to you via email following this presentation. Please take this opportunity to let us know which topics you would like to see in future ISCT webinars. And completion of the evaluation will allow you access to the webinar recording. Thank you very much.